Hello everyone. This is Dr. Anisha from St. Albert's College, Autonomous, Ernakulam. Today we will be learning something about the basics of plant tissue culture. So, as we all know, plant tissue culture has developed as a very versatile tool in the field of plant sciences. This particular area has diversified so much and finds application in a number of areas, both commercially as well as at the level of research. So, what is actually plant tissue culture? So, we all know plant tissue culture is growing plants in conditions which are not natural, or for a definition, the in vitro cultivation of plant cell or tissue under controlled conditions to produce new intact plants. That is what tissue culture is. So here what we try is, normally plants are grown in soil from their regular propagules, which may be seed or cuttings or otherwise, or a number of things are available. So instead of that, we try to grow plants under an artificial set of conditions, and we try to multiply them. We try to get new plants in those artificial conditions. That is plant tissue culture. So the plants produced through tissue culture, since they are being raised from the vegetative parts, these plants will be genetically similar to the parent plant. Whatever parent plant material we are using, the genetic makeup of the new plants will be similar to those parent plants. So it is widely used these days, not just for multiplying plants, but also for a number of other purposes, where it fares very nicely. Going to the history of plant tissue culture, how this field of study came into existence. So the things go back till the year 1902, when a German scientist called Gottlieb Haberland, he actually was trying to grow plants under an artificial set of conditions, and he could culture fully differentiated plant cells which were isolated from different plant types. Because of this fact that he started this area of study, he is also referred to as the father of plant tissue culture. In 1943, the first book on plant tissue culture was published, and it was authored by White. In 1962, which is a very remarkable year as far as plant tissue culture is concerned, because it was in this year that the very famous and universally used medium, MS medium, or more correctly, Murashige and Skoog medium, that was formulated. It was formulated by scientists Murashige and Skoog, and today MS medium is the most extensively used plant tissue culture medium. It is the most popular medium, and it usually gives very good results for most of the plants that are being cultured. In 1964, which is a remarkable year in the field of plant tissue culture in India, Guha and Maheshwari, they produced first haploid plants using pollen grains, and it was anther culture. They made use of pollen grains from Datura. In 1972, Carlson produced the first interspecific hybrid of Nicotiana tabacum, and he used the technique of protoplast fusion, which also finds multiple uses in research also these days. In 1977, Chilton et al. He integrated the TI plasmid DNA that comes from Agrobacterium tumefaciens in plants. Year 1981 saw the new terminology somaclonal variations that was given by Larkin and Scowcroft. 1984, Horsch et al. Developed transgenic tobacco by transformation using Agrobacterium. In 2005, the rice genome was sequenced under the International Rice Genome Sequencing Project, and it made use of rice tissue culture extensively. In 2006, IAPTC and B, it was renamed as International Association of Plant Biotechnology, IAPB. So, plant tissue culture has grown as a very versatile field of study, and it finds application in a number of areas that range from clonal plant propagation, germplasm storage, production, and maintenance of disease free plants, and also as a valuable adjunct to the conventional methods of plant improvement. So, plant tissue culture has become a very necessary or very significant component of plant based research these days. Now, what makes plant tissue culture work? What is the basis of plant tissue culture? So, there are certain inherent properties seen in plant cells that makes tissue culture happen. And the most significant of those properties is totipotency. Totipotency, the inherent potentiality of a plant cell to give rise to a whole plant, is actually totipotency. It is a capacity which is often retained even after a cell has undergone final differentiation in the plant body, and it is sometimes also referred to as cellular totipotency. So, even if the cell is in a fully grown plant, it may be able to give rise to a whole new plant by itself. That this property is inherent in plant cells, which is made use of in doing tissue culture. During when this totipotency works out, two major phenomena are seen to happen. One is dedifferentiation and the other is redifferentiation. The two terms are very descriptive of what they actually mean. Dedifferentiation is the phenomenon of a mature cell reverting to the meristematic state and forming undifferentiated callus tissue. The cells in a differentiated tissue, they revert back to the meristematic state and generally give rise to a callus formation. Redifferentiation is just the reverse. Here, it is a process where the cells divide and produce cells which once again lose their dividing capacity but mature to perform specific functions. So, dedifferentiated cells, when they undergo differentiation again, this phenomenon is referred to as redifferentiation. So, altogether, these two phenomena leading to the totipotent capacity of plant cells helps in plant tissue culture happening. What are the requirements for doing plant tissue culture? How should we approach this technique? So, primarily, there are three major requirements for doing plant tissue culture. First is aseptic conditions. It is a prerequisite for doing plant tissue culture. Aseptic conditions are a must for conducting plant tissue culture. Next is controlled environment. As I mentioned in the very beginning, plants are grown in conditions which are different from their natural growing environment. So, whatever is the artificial condition, it has to be controlled. All the parameters have to be given in a controlled manner and it has to be ensured at all points of plant tissue culture. Appropriate culture medium. Plants are provided with a culture medium which contains macronutrients, micronutrients, vitamins, other organic components, growth regulators, carbon source, and some gelling agents. So, this is something mandatory to be given for a plant for its growth because nutrition is of prime importance. These three are the prerequisites for doing plant tissue culture, and all the parameters coming under these three have to be taken care of with due care so as to ensure that the plant tissue culture is successful. So, we will take each one separately. First was aseptic conditions. So, aseptic conditions are ensured by conducting different type of sterilization procedures. So, why do we need to go for a sterilization procedure? We use medium for doing plant tissue culture and this medium contains rich nutrients. So, nutrients, they support plant growth, but at the same time they also support the growth of other microbes like bacteria and fungi. So it is all possible that when we use the nutrient medium, the medium is used by bacteria and fungi for their growth. And the sad part is, if there is an entry of a bacteria or a fungi in the nutrient medium, it will be using the medium faster than the plant tissue that we have used. So the growth of the fungus or bacteria will be faster than the plant, and ultimately the plant will be killed. So we won't get the plant culture, we will end up with a bacterial or a fungal culture. And there is also possibility that these microbes, they release toxins into the medium. So that's why sterilization is of utmost importance in plant tissue culture, because we don't want any kind of microbes growing in our cultures. We only want the plant to grow. So it is of great significance that sterility is maintained at all points of tissue culture. Everything that is used for during the procedures of plant tissue culture, they have to be sterilized. The procedures that will be followed will vary with the item that has to be sterilized, but everything has to undergo a sterilization procedure, be it labwares, glasswares, medium, the plant material, or even the person who is doing the plant tissue culture. That is regarding sterilization. Now, coming to the artificial growth conditions, normally the cultures will be maintained in an incubation room or a growth room, where we provide the artificial conditions for growth. So, there it is mandatory that the growing conditions are properly maintained, optimum growth conditions are properly maintained, so that the growth and multiplication of the explant is happening in a proper manner. The cultures, after being inoculated, they are incubated in a growth room. So, in this growth room, temperature, humidity, light, all these parameters have to be taken care of with due seriousness. Temperature, the temperature normally that is required in plant tissue culture conditions is something around 25 plus or minus 2 degrees Celsius, which is generally maintained by air conditioners. Then light intensity. Generally, cool white light is preferred, which should be in the range of 3000 to 5000 lux. Then relative humidity, which is generally maintained above 50%. And if there are variations, then there have to be interventions done to ensure the required type of humidity in the cultures. Then shelving units to maintain cultures in a neat and tidy environment. So these are the conditions with respect to the growth or incubation of the cultures. Now the last one is plant tissue culture medium, which is of utmost significance as far as tissue culture is concerned, because we put the plants directly in the tissue culture medium. So the medium composition is something that has to be taken care of with due seriousness. The growth, development, and morphogenic response of an explant in culture depends on its genetic makeup, surrounding environment, and composition of the culture medium. So, selection of the right type of culture medium having the right composition is very important. Murashige and scoop medium, it is commonly used for doing plant tissue culture, though there are other more specific medias also available for certain plants. Whenever we are making use of a plant tissue culture medium, generally plant growth regulators or plant growth hormones are also added to get the required kind of response in the cultures, and that will depend upon what is the objective of a tissue culture experiment. Generally, the plant growth regulators that are being used include auxins and cytokinins, and also sometimes there may be gibberellins which are also used. These are the most commonly used ones. So these three components, the aseptic conditions, then the optimal growth conditions, and finally the culture medium. These three form the very essential prerequisites for doing plant tissue culture. I hope these basic concepts regarding plant tissue culture are clear to all of you. Thank you.